There are many types of biosensors, and this is because they can differ in the bioreceptors they use, or the transducer signals, or even in their application. Now, in this video, we'll be looking at two of the four main types of biosensors across all these categories. These are calorimetric, fluorescence, surface plasma resonance, and antigen-antibiotic interaction, and today we'll be looking at the calorimetric and fluorescence biosensors. First, let's start off with calorimetric biosensors. What are they to start off with? Well, calorimetric biosensors detect changes in light absorption or heat generated by biochemical reactions. What does the term calorimetric mean? Well, calorimetric, derived from the word calorimetry, means the process of measuring the amount of heat that's released or absorbed during a chemical reaction. The term becomes more easier to understand when tying it with the term calorie, as in burning calories, for instance. Now, the word is derived from the Latin term calor, meaning heat, and this is also the same root word for calorimetric biosensors. So then, how do these calorimetric biosensors work? Well, they work by the following mechanism, and let's approach this with the example of glucose level detection. First, the biosensor containing the bioreceptor reacts with the target analyte. Now, in this specific case, this would be the glucose, and upon interaction with it, it generates a reaction. Now, the interaction between the analyte and the bioreceptor triggers a chromism or an endothermic or an ectothermic reaction, which essentially means that these reactions result in a change in color or heat. In the case of glucose detection, the bioreceptors, the glucose oxidase, an enzyme that's capable of catalyzing glucose, would then catalyze the reaction of glucose with oxygen and produce gluconic acid and hydrogen peroxide. Now, the chemical equation of the reaction that takes place here are as follows. This is glucose. As you can see on the reactant side, we have glucose and oxygen, and the enzyme glucose oxidase catalyzes the reaction, which produces gluconic acid and hydrogen peroxide. Now, this reaction is an oxidation reaction, and it is exothermic, meaning that it releases heat as product. Now, the amount of heat that's released is proportional to the concentration of glucose in the sample. The higher the glucose levels results in more significant heat generation. The sensor then incorporates a thermistor, which would then detect the temperature changes caused by the reaction. This allows us to understand by more quantitative means the changes in glucose levels. Now, next are fluorescent biosensors. Fluorescent biosensors are analytical devices that detect biomolecules present in biological samples by using fluorescence, which occurs when electromagnetic radiation is absorbed by fluorophores or fluorescently labeled molecules. Fluorescent biosensors work in a similar way to calorimetric biosensors. To explore the working mechanism, we'll be looking at an example of fluorescence biosensor for virus detection, an example from a recent study in the Journal of Nanobiotechnology 2021. So, as a more pretext to the detailed explanation of the working mechanism, the fluorescence biosensor was used for detecting the SARS-CoV-2 virus, a pathogen that's responsible for COVID-19. Now, the biosensor uses quantum dots as fluorophores, which are essentially semiconductor nanoparticles that can easily be triggered and have stable fluorescence properties. Now, in the case of this specific example, the bioreceptor was a modified DNA strand that binds specifically to the RNA of the SARS-CoV-2. The analyte, as you can probably would have guessed, is the SARS-CoV-2. Now, the way that this worked was there were quantum dots functionalized with the DNA strands, and once the virus's RNA was detected, the DNA binded to it, which caused a measurable change in the fluorescence due to the quantum dots. The quantum dots emit fluorescence that is directly proportional to the concentration of viral RNA present. This means the more brighter it shines, the higher the concentration of viral RNA. The emitted light was then captured by a photodetector, which quantified this qualitative observation of light emission. And that brings us to the end of our video. Thank you for watching, and in our next video we'll be talking about the other two biosensors.